Greetings, everyone. This is Oscar Segurado. I'm the Chief Medical Officer of ASC Therapeutics. This is a company, a biopharma organization based in the San Francisco Bay Area. We are working on 3 lines of therapies. First of all is gene editing with CRISPR-Cas9 for hemophilia A, replacing factor VIII. Using also the same transgene, we are also working on gene therapy. And the third line of work, which I'm going to be focusing today, is on allogeneic cell therapies. And I'm going to describe several aspects of what we are doing, which are very relevant to this conference. And I think that I will also focus on an aspect that is very relevant of the work that several biopharmas are doing. And this is especially on related to personalized and precision medicine. So let's start going for it and going first with an outline of my presentation, starting with the immunoregulatory power of allogeneic cell therapies. And what I'm going to do today is to ensure that I provide you the basic information you need to ensure that you can understand all the data that I'm going to put in front of you. I'm going to simplify that. I'm going to really try to make sure that you get the high level picture of everything that we're doing and see the impact that we're having on several rare diseases, which is the focus of this presentation too. Let's start with the outline in terms of the immunoregulatory power of allogeneic cell therapies. I'm going to talk about the intended use, and I'm going to describe to you what is graft versus host disease. Then I'm going to talk about our therapeutic modality, which is what type of cells are we using, these off-the-shelf cells, which are Decidua Stromal Cells. And then I'm going to discuss these immune biomarkers, the way we are able to take a look into different parameters, different aspects of the way this drug interacts in terms of pharmacokinetics, PK, and pharmacodynamics, PD. Then I'm going to go into summarizing all this information into what does it mean from a personalized medicine aspect for allogeneic cell therapies, and ensuring that I talk about biomarkers for patient selection and biomarkers for patient monitoring. Let's go for it. Let's start with the first section with the immunoregulatory power of allogeneic Decidua Stromal Cells. And let's start with the indication, acute graft versus host disease, GVHD. And first of all, let's realize that many patients with different types of blood cancers and several other conditions require an infusion of cells from another donor. These are called allogeneic hematopoietic stem cell transplants. And these happen regularly all around the world, about 30,000 allogeneic HSCTs per year worldwide. And what happens occasionally is that there is a reaction of these immune cells that are infused into the patient are going to not only do their job, which is kill the cancer cells or the cells that really have this condition, but also potentially being deleterious to the patient. So up to 30 to 75% of patients receiving allogeneic HSCT, hematopoietic stem cell transplants, develop acute and chronic graft versus host disease. We're going to focus here on acute graft versus host disease. And there is a significant transplant-related morbidity and high mortality rate in these patients, especially those that achieve what is the most serious cases, grade 3 and 4 of acute graft versus host disease, and especially those that do not respond to the standard therapy, which is steroid therapy. These are called steroid refractory graft versus host disease, and these are the ones we're going to focus on. So steroid refractory graft versus host disease is considered a rare disease. Now, what happens when you have this reaction? These graft, these donor cells that are attacking the body. The first thing is that there is already a basic initial damage done to several tissues in the individuals, because they need what is called a conditional treatment. A conditioning treatment means that they need to be immunosuppressed. Their immune system is basically wiped out, so that the donor cells are going to replace, and you're going to get a kind of new immune system. So despite the importance of making sure that there is compatibility, and this is through HLA and other aspects, where really bring these cells relatively associated to the patient, we also see that there is also another second phase, which is the donor T cell activations. These T cells should do the role, again, wiping out the cancer cells, but in some cases, they are also activating the entire immune system that has been transplanted to the patient, and this goes through so-called Th1 or T helper cells. The third step is that there is an immune-based host immune tissue damage, and this is when there is a target cell lysis, and we're going to see now which tissues are mostly affected by this therapy. So now let's go into the other basic concept that I want to make sure that you understand. And this is our allogeneic cells, which are called Decidua Stromal Cells, DSCs. Let me explain you what that is. When a woman is pregnant and delivers, there is obviously the fetus, but the fetus is also protected by a number of layers. They are called decidua, chorionic membranes. There is also the amniotic fluid, the Yolk sac, and what we found out based on work conducted at the Karolinska Institute in Sweden is that there are some cells of maternal origin. They are obtained from the discarded placentas after the delivery, and these cells, once characterized, isolated, and expanded, have potent immunosuppressive effects through the inhibition of T cells and other immunoregulatory characteristics. So these, once we see that these cells can be manufactured in a consistent manner, we are able to also reduce the manufacturing cost, so that these allogeneic cells from a single placenta can treat from 100 to 200 patients. I want you to realize that we're talking about off-the-shelf cell therapies. You probably have heard about CAR T therapies, these chimeric antigen cells that are used to treat also blood cancers. But what we have here are cells that are basically frozen, thawed when they are given to the patient, and they have been already through a number of regulatory aspects which show that they are accepted by the agencies. Now, these Decidua Stromal cells were developed originally by a group in Karolinska Institute, which has been publishing a number of papers, starting, you can see here, in 2012, over 10 years ago. And there are papers talking about the immunosuppressive effect, their effect on the regulatory T cells, the fact that they are derived from the placenta, the fact that there are certain immunosuppressive properties of these cells that they can also they have also worked on the distribution of these cells. There is preclinical toxicity evaluation. All this information serve us to decide to in-license this asset. These cells from the Karolinska Institute, we worked very closely with them over the past 5 years, and we decided to develop them in the United States and ensure that we were presenting all this data to the regulatory agencies. Based on all this work that we have been doing over all these years, we were able to publish a review of all these data that was generated by them, also by us. because we were also, and we are going to see in a moment, able to present all this data to the FDA. So they show that all the data, all the work that was conducted in an academic setting in the Karolinska Institute could be used to justify that we were moving directly to a phase 2B clinical trial. You're going to see that in a moment. And this is the publication that is right now in press. It was accepted just a few days ago. That's why you see here this kind of messy overview with a number of questions. These are galley proofs. And the title is Novel Therapies for Graft versus Host Disease with a Focus on Cell Therapies. And you see here the authors. The first three are the authors that have been working on this asset for this long time. And then you have our team at ASC Therapeutics. If you want to access, for now is the abstract. In the next few days is going to be the entire publication is already in the internet. So here are our distinguished advisory board that have been working with us over the past several years. Robert Sizer, he is the leader of the field of graft versus host disease. He's the first author of a number of publications using JAK2 inhibitors, ruxolitinib. Then Professor Ole Ringden and Professor Benham Sadeghi both have been working at the Karolinska Institute and working very closely with us, doing what is called a tech transfer. So all the information, all the work that they have been doing has been translated into the way we can now not only identify, characterize, and isolate these cells, also to expand them and manufacture them. We have a manufacturing facility here in the United States. And then we also identified 3 top leaders in the field of cell therapies for graft versus host disease. And this is Professor Negrin in Stanford University, Professor Wagner, and Professor Spall. So this is the critical work that we help us consider adopting this technology and bringing it into the United States and ensure that we go through all the regulatory process, first in the United States and then in Europe. In this graph, you see the overall survival of patients with acute steroid refractory graft versus host disease that have been treated with different modalities. You have in the vertical axis the cumulative survival, the overall survival. In the horizontal axis, you have days after the therapy. And you can see at the bottom the historical controls in blue, where you can see that basically immediately these patients have a very high mortality rate, which practically happened in the first 200 days. And this basically, obviously, is this flat line. Then there is another modality that has been used and very, very thorough assessed, even by this group of Karolinska Institute, which is the bone marrow mesenchymal stromal cells. These are similar to the Decidua Stromal Cells. And this group in Karolinska had been working originally with these cells for many years. And they are the ones that decided to, although there was an improvement, as you can see here in the green line, from the historical control to the green BMMSCs, there is an improvement in survival. They decided to move into the Decidua Stromal Cells, this treated group, which shows a much better overall survival. And you can see that that's apparent already in the first 200 days, and is also maintained over time with the red line. And definitely this is work that has been published, and it was original data conducted in an academic setting. This was presented to the FDA, and the FDA accepted that to be able to move into a more advanced clinical trial. Now, this asset in our hands is now called ASC 930, and it has received a number of regulatory designations by the FDA and the European agency EMA. And we have received the IND clearance from the FDA to conduct a phase 2B clinical trial. So based on the data from the Karolinska Institute, data also generated by our team, we were able to put together a package of over 4,000 pages of documents that were submitted to the FDA, and the FDA gave us the green light to start with the phase 2B clinical trial that we are right now involved with. We also obtained from the FDA orphan drug designation. What this means is that we get a tax credit. We are able to have exclusivity for a number of years. And this is also something that is provided to diseases that are considered an orphan disease or a rare disease. So that's why we are now able to conduct a single-arm, non-randomized, and open-label study. Now, this is the clinical trial design, and, which was presented to the FDA and we are conducting. And here you can see also that there is a link at the top. You can find that in clinicaltrials.gov, and you can see how we are planning to do this study. Also, all this was in conversation with the Karolinska team, with our advisory board members. And the idea is that we are going to take patients first that show an acute graft versus host disease. We're going to follow these patients for the first week, and we follow a number of criteria that indicate that these patients are not responding. And when these patients are not responding, they're going to get our first course of treatment. We hope that the majority of the patients are going to respond in the first treatment. If not, and after the primary endpoint, that is overall response rate at day 28, then they are going to get a second treatment course, and we're going to have a follow-up that is going to be up to around 6 months. Now, let me now move on and start talking about the mechanism of action. So this is absolutely critical to understand why these cells are working, and also the way we are going to monitor in our clinical trial how these cells are impacting the immune system. So we're going to first take this population of a steroid refractory acute graft versus host disease. We're going to look into the impact on the immune system by immune profiling the actual cells that we are infusing, and also the profiling the alloreactive T cells of the individual, the way we see also the profiles for cytokines and adhesion molecules, and we're going to do an entire profiling of all other immune cells. So let's talk about these immunomodulatory effects of Decidua stromal cells. And there is also a publication where we have looked into the type of biomarkers that I'm going to describe to you. But again, as a summary of what I've already indicated in previous slides, we take these Decidua stromal cells that are derived from the mother, also protecting the fetus, and they have a positive effect in two ways. First, they are going to increase and upregulate the checkpoint inhibitors, PD-1 and PD-L1. PD-L1, PD-L2, and these checkpoint inhibitors are the ones that you sometimes want to decrease in patients with cancer. But if in terms of an immunoregulatory perspective, we want to increase them. And also we also see an increase in regulatory T cells. So we're going to immune regulate the entire system of these patients that is failing. And what we do from a negative perspective is that we are going to control inflammation in these patients, and the majority of these patients are going to see inflammatory conditions in the skin, in the gut, also in several other organs of the digestive system, like the liver. And we're also going to see an apoptosis of alloreactive T cells, so the cells that are actually from the donor and are causing all this damage. Now, here are some examples. Don't worry about these very complex graphs. We have a lot of data, and I just want to give you a sense about how we are able to identify the T cell regulation by the Decidua stromal cells. You can see here in these graphs how you always have a bar, which is the first one, and the second one with 2 stars, meaning that there is a significant decrease of immunosuppression when you use the MLR, which is mixed lymphocyte reaction, plus several types of adjuvants, and we can see that in all cases we see the effect of the DSCs being extraordinarily significant. Now, again, don't worry about the graphs here, the bar graphs, but the point here is that we also see regulation of cytokines and adhesion molecules by the DSCs. And at the top, we can see how there is a regulation of the cytokines that are released by alloreactive stimulated T cells. We also, again, you can see that there is a bar at the, and then a second bar where there is a significant decrease. That's the only point that I want to make here. At the bottom, we see the expression of high levels of adhesion markers, which are a special aspect of these Decidua stromal cells. Again, this is scientific data. I don't want you to go into details. Obviously, all this has been published. And I want to give you now a sense about what we are planning to do, what we are already doing with patients that are entering our clinical trial. We have identified a company that is using mass cytometry, and they are using what is called the TOCU panel, and which captures the entire immune system. We look into a number of markers, surface markers of the T cells, NK cells, myeloid cells, and other cells in the immune system in the whole blood. So we get a complete profiling of the immune system, and we are going to check how this profile changes over time in these patients. And again, this is in collaboration with Tycho. We also definitely need to understand what is the impact of our cells on controlling the immune effect that we're seeing with graft versus host disease in tissues and organs. So we obviously need to understand the safety and the efficacy of our therapy. And something extraordinarily important is to ensure that we understand what are the pharmacokinetics of the Decidua stromal cells, how they behave when they are infused. And this is original work from Karolinska Institute in a number of patients where they were looking into how these cells work. And what we could see is that within hours, the cells initially go into the lung, and then they start deriving into the liver and the spleen. And basically, after 48 hours, these cells, these Decidua stromal cells, disappear. So that's why we can see that this immunoregulatory effect can be called a hit and run type of effect. So this is not like CAR-Ts where you have the effect that takes place over weeks, months, and they control all these kind of cancer, because this is an acute condition. It's very important that these cells just do their job, do the immunoregulatory role, and then basically disappear. And eventually we can treat these patients again. And that's what we do in a weekly basis, in a first course, and potentially in a second course. Now, very important too is to understand the pharmacokinetics of these cells. And for that, we are working with another company here in the Bay Area, San Francisco Bay Area. It's called CareDx. And they are able to take a look into how these allogeneic cells are working over time. And what we see with their technology, with their product called AlloCell, is that they're able to assess several aspects that other technologies like quantitative PCR are not able to do. Again, this is a collaboration that is going to serve us to select and to monitor patients. And with all this information, I would like to condense that into how we see that applying to the entire field of cell therapies, and specifically to allogeneic cell therapies. So all this learning, everything that we are doing with this approach, with this revolutionary type of asset that potentially is going to be not only relevant for patients with graft versus host disease, but also other types of immune dysregulation, we are looking into what we call personalized medicine in allogeneic cell therapies. So we are bringing to the table a number of biomarkers that are going to be help us to first select patients who may benefit from a cell therapy. And because maybe some of the patients, based on these biomarkers, may not be adequate for this type of therapy. So we're going to characterize and phenotypes these allogeneic cells. I told you how we are going to do that. We are also going to take some of these cells and look and into ex vivo functional effects on target systems and organs. There are also organoids that could help us to do that. And then for the monitoring of the patient response over time, we want to make sure that we distinguish efficacy biomarkers versus safety biomarkers. And those are all obviously going to be extraordinarily relevant when we present our data to the regulatory agencies, because they want to see, again, if these cells are having the effect that we expect in terms of clinical activity, and also that they don't bring any kind of risk that goes beyond what we is expected on this type of cells. So these are, that's why we are considering, and we are going to work on efficacy biomarkers and safety biomarkers. So with all of that, we are really bringing to the table a targeted therapy that is based on biomarkers that provide information essential for safe and effective use of the corresponding therapeutic product. And there is also a big movement now around the entire biopharmaceutical industry looking into what is called omics data. We're going to incorporate many of those, including genomics, transcriptomics, epigenomics, proteomics, phosphoproteomics, metabolomics, and we're going to move from just single biomarkers that I've been discussing before to having signatures that are going to correlate and combine some of these biomarkers, networking them, ensuring that we get any kind of either machine learning or generative AI approaches, so we're able to target and personalize our therapies to the patients that are going to really benefit from them. So with all of that, we're moving from a traditional one-size-fits-all approach that have been mostly embraced by the pharmaceutical industry until now, for a more personalized medicine approach, which is going to be based on a genetic profiling, on also biomarker profiling, distinguishing patients that receive the targeted therapy versus those that could receive other type of standard therapies. So in summary, this is why we consider ASC 930 as a cutting-edge allogeneic cell therapy. There is only right now one product approved. It's called ruxolitinib, a JAK2 inhibitor, but it's limited to patients over 12 years of age. ASC 930 has the potential to be approved, the first approved cell therapy for all ages. The Decidua Stromal Cells are related to mesenchymal stromal cells, but definitely much better, much potent immunomodulatory activities. And we think that there is a big market opportunity because graft versus disease will reach market value of over $5 billion in a few years. So the competitive edge is, again, this immunosuppressive effect that we are obtaining through these stromal cells, excellent efficacy and safety profiles, solid clinical data that surpasses everything published, including ruxolitinib, this JAK2 inhibitor. And finally, the fact that we have a robust cell therapy manufacturing process with our experienced CMC partner, at this point, City of Hope, and we have already an IND clear. So the FDA and the EMA have provided us also a number of regulatory designations that also bring us to the potential of showing activity in this indication and then moving into other indications. So the take-home message is that placenta-derived Decidua stromal cells are a novel allogeneic cell therapy which precisely target patients with severe acute graft versus host disease. But we also see, and once we are able to figure out everything related to the activity, mechanism of action, and manufacturing aspects of these cells, we plan to bring these cells to other potential indications, including autoimmune and acute inflammatory disorders. With that, I would like to thank you for your participation, for your interest. And if you have any questions to me, since I'm not able to address that live, I would be happy to respond to them. And here you have my email address. It's just my name, oscar.segurado@asctherapeutics.com. And with that, thank you so much for your attention.